Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Imagine your brain. It's protected by, well, one of the body's most amazing defenses, the blood-brain barrier. Think of it like this incredibly selective border patrol, right?
Yeah.
Designed to keep threats out.
It really is remarkable. A complex structure of specialized cells, tightly knit together, controlling almost everything that tries to get into the brain's very delicate environment.
Okay, but what if something nasty, something persistent, and frankly deadly gets through that wall? We're talking today about tuberculosis TB and a really severe form that attacks the brain itself. It's called tuberculous meningitis.
Yes, and it's a very serious complication. Tuberculosis usually hits the lungs, as most people know, but in somewhere between maybe 1% and 10% of cases, the bacteria find their way to the central nervous system. And tuberculous meningitis is, unfortunately, often fatal and really quite hard to treat.
And we also know that some people are just at higher risk. People living with HIV, for example, seem to be much more vulnerable to this severe brain infection. That co-infection piece makes things really complex.
It absolutely does. And for the longest time, it wasn't totally clear how the tuberculosis bacteria, Mycobacterium tuberculosis, actually managed to cross that blood-brain barrier. The main idea floating around was the Trojan horse theory bacteria hiding inside immune cells that cross over.
Right, the classic Trojan horse. But is that the whole story? Can the bacteria maybe find their own way across directly? And that's really the core question in the research we're digging into today. It's a recent study, a fascinating one, from the Crick Institute, published in the Journal of Neuroinflammation.
Yeah, this team used some pretty advanced lab models to get right at that mystery. How do these bacteria breach the barrier? And crucially, what difference does it make if someone also has HIV?
So our mission in this deep dive, custom made for you, is to unpack this study. We want to explore how these bacteria might break through the brain's defenses, possibly in ways we didn't fully appreciate, and how HIV might, well, play a role in that whole process at the cellular level. So let's jump into the study itself. What were the researchers really trying to figure out? What was the main aim?
Well, their primary goal was to watch, really closely at the microscopic level, how Mycobacterium tuberculosis interacts with the specific cells that make up and support the blood-brain barrier. They wanted to see if the bacteria could actually get across, and very importantly, how HIV infection might influence things.
Okay, so how did they tackle that? I mean, building a blood-brain barrier in a dish sounds, well, tricky.
It definitely is complex. But they used some clever methods. They didn't try to build a whole brain right away. First, they isolated the key players, the individual cell types. You've got the endothelial cells lining the blood vessels, the parasites and astrocytes wrapped around them, and the microglia, which are kind of the brain's own immune cells sitting just behind the barrier.
Ah, so they broke it down cell by cell initially.
Exactly. They exposed each cell type individually to the tuberculosis bacteria. And what they found was pretty striking. The bacteria could get inside and actually grow and multiply within all of these different cell types.
Wow. So these brain barrier cells aren't just passive gatekeepers. They can actually become hosts for the bacteria.
That's a really crucial point, yes. These cells can become a niche for the bacteria to replicate. Then they added the HIV factor. Sometimes they'd expose cells to HIV before the tuberculosis bacteria to see if HIV sort of primed the pump for bacterial infection.
But the really innovative part, reading the source material, seems to be that 3D model they built, right? Actually replicating the barrier structure.
Oh, absolutely. That was key. To go beyond just single cells and mimic that layered defense, they built this sophisticated 3D system. They used a sort of porous membrane scaffold. On one side, the blood side, they put the endothelial cells. On the other brain side, they added parasites and astrocytes, and they included microglia nearby too.
Okay, that sounds much closer to how it might work in the body.
It allows them to look at movement across those layers, not just infection of a single cell type. So using this 3D model and their single cell work, they measured several things, like how easily stuff could leak across the barrier, its permeability. They checked if the cell layers stayed intact, structurally sound. And the big one, could the tuberculosis bacteria actually get from the blood side to the brain side in their model?
Did they also look at, what the infection was doing to the cells? Were the cells getting stressed out?
Yes, that was vital. They looked for signs of cellular damage. They measured things like reactive oxygen species. Those are molecules that signal cell stress and injury. And they tracked levels of glutamate, which is a brain chemical messenger.
Mhm.
But if too much of it builds up outside cells, it becomes toxic over exciting neurons.
So it's not just breaking through physically, the infection might also be creating a kind of toxic chemical mess right at the barrier.
That's exactly what the data suggested. And they also found something interesting with astrocytes, those star-shaped support cells in the brain. The infection seemed to make them become neurotoxic, meaning they shifted into a state where they could potentially harm nerve cells. They saw increases in markers linked to this harmful state.
Okay, that paints a pretty disruptive picture. Let's get into the nitty-gritty of the findings. What happened when the tuberculosis bacteria met these cells and the barrier model?
Well, confirming the early tests, the bacteria didn't just get into the cells. They really settled in and multiplied over time.
And the barrier itself, did it stay strong?
This is where the idea of a direct attack really comes into focus. They used a system to measure the electrical resistance across the cell layers, basically, how tightly sealed they were. Over about 80 hours, the tuberculosis infection caused a massive drop in the integrity of all the cell types they tested. We're talking over an 80% decrease in stability.
80%, wow, that's huge. That suggests the bacteria are really doing some serious damage to the structure. It's not just sneaking past.
It tells you they're actively undermining the very cells forming that barrier. And this played out clearly in the 3D model. Tuberculosis infection made their model barrier significantly more permeable, leakier after only 2 days. And it got even leakier by day 6.
So the bacteria punch holes, effectively, and did that let them cross?
Yes. That increased leakiness directly correlated with Mycobacterium tuberculosis bacteria successfully passing across the barrier model in the lab. They could actually find viable bacteria on the brain side of their setup.
Direct proof of crossing in their system. And you mentioned the cell stress signals. What were those again?
Right. They saw the production of those reactive oxygen species, clear sign of damage. And that buildup of glutamate outside the cells, which is potentially toxic to nearby neurons, it really underscores that the breach isn't just physical, it's creating a harmful biological environment right there.
And the astrocytes turning neurotoxic. So the brain's own support crew could turn harmful.
Potentially, yes. It suggests their normal supportive role gets disrupted, shifting towards something that could add to the damage within the brain tissue itself.
Seems like these bacteria have a multi-pronged attack strategy. Did the researchers figure out how the barrier got so leaky? Was it like breaking the seals, the tight junctions between cells?
That was one of the really interesting parts. Even though the barrier became much leakier, the bacteria didn't seem to drastically reduce the amount of the main tight junction proteins, the primary zippers holding cells together. They did note a slight decrease in a different type of adhesion protein, though.
That seems weird. If the main seals aren't broken, how does it get so leaky?
Well, this points towards a really important idea. It suggests maybe the bacteria aren't only squeezing between the cells. Through those junctions, that's the paracellular route. The fact that they also get inside the barrier cells and grow there suggests another route. They might be going through the cells themselves, the transcellular route. The damage they cause inside the cells could weaken the whole structure, facilitating passage both between and through.
Ah, okay. So they're not just jimmying the lock. They're also weakening the door itself, maybe allowing passage right through it.
Exactly. They're messing with the cell's internal integrity. Okay, so now let's bring HIV into this picture. How did that change things? This is where the study really highlights that increased risk for people with HIV. What they found was that HIV co-infection actually boosted the initial entry of the tuberculosis bacteria into several of those key border cell types, the astrocytes, parasites, and microglia in the single-cell tests.
So having HIV around somehow makes it easier for the TB bacteria to invade these critical gateway cells.
It looks that way. It seems to prime them, making them more vulnerable to the bacteria. And wasn't just getting into HIV, also increased how fast the tuberculosis bacteria grew once they were inside the parasites and endothelial cells.
So more bacteria getting in initially, and then they multiply faster once inside. That really dials up the threat right at the gate, doesn't it?
It certainly seems to. And they saw something else really intriguing, what they called a bystander effect. It seemed like tuberculosis bacteria were more successful at infecting cells that were simply exposed to HIV, even if those cells weren't actually infected by HIV itself.
Whoa, that's wild. Just the presence of HIV nearby, even indirectly, makes the environment better for the TB bacteria.
It suggests HIV might be changing the local environment in subtle ways, maybe releasing signals that somehow benefit the tuberculosis bacteria. Now, interestingly, in the 3D model, HIV didn't seem to make the barrier even more leaky than TB already did. But, and this is critical, HIV co-infection did increase the total number of tuberculosis bacteria that managed to cross the 3D barrier, especially early on, after 2 days.
Okay, wait. The barrier wasn't leakier overall, but more bacteria got through. How does that work?
The thinking is, because HIV helped more bacteria get into those border cells, the endothelial cells of parasites, and grow better within them, there were simply more bacteria right there at the barrier, poised to cross. So even if the leakiness rate wasn't higher, having a larger population of bacteria at the starting line meant more ended up crossing, whether they went between the cells or through the damaged cells.
Got it. It's like HIV isn't necessarily blowing bigger holes in the wall, but it's helping muster more invaders right at the breach point.
That's a good analogy. And they also saw that this bystander HIV effect slightly worsened the tendency for astrocytes to become neurotoxic when TB was present. Plus, HIV slightly increased the release of certain inflammatory chemicals, cytokines like interleukin-1 beta, interleukin-6, interleukin-8 from the cells in the 3D barrier model. That inflammation itself could cause further problems.
This study really is unpacking a lot of complexity here. So let's try and synthesize this. What are the big takeaways, the major implications?
I think one of the biggest is that this provides pretty solid lab evidence that Mycobacterium tuberculosis can directly attack the blood-brain barrier. It does this by actively damaging and weakening the cells that form that barrier.
So it gives strong support to the idea that it's not just the Trojan horse mechanism, although that might still happen too. These bacteria have a direct assault strategy.
Precisely. It offers a convincing alternative. Or maybe it's complementary. The bacteria can be direct invaders attacking the border head-on.
And this links directly back to why people with HIV are more vulnerable.
Yes, exactly. This work helps explain why, at a cellular level, HIV increases the risk for tuberculous meningitis. It looks like HIV makes those key barrier cells more susceptible to invasion and growth by the TB bacteria. This potentially makes it easier for the bacteria to get into the brain. As Dr. Robert Wilkinson, one of the senior researchers, mentioned, this helps unravel that known clinical risk.
And understanding how they get in, that seems absolutely fundamental if we want to find better ways to stop this disease or treat it.
It really is. Elise Proust, the lead author, made this point very clearly. Knowing this mechanism could open up new treatment avenues. If we know how they're getting in, maybe we can design drugs to block that specific process. Or, here's an interesting thought she raised. If the infection makes the barrier leaky for a time, could we maybe use that leakiness to get drugs into the brain more effectively when they're needed most?
Using the damage caused by the bacteria to sneak the medicine in.
Potentially. If we could figure out that window of increased permeability, it might help overcome the big challenge of getting drugs into the brain, which, as Dr. Wilkinson also stressed, is notoriously difficult, especially when you're dealing with co-infections like TB and HIV.
It's fascinating research, but like any study, especially using lab models, there are limitations we should probably acknowledge, right?
Oh, absolutely. And it's important. The main one is that this was all done in vitro using cells and simplified barrier models in dishes. While that's great for isolating specific interactions, it just can't fully capture the incredible complexity of a living brain and the body's entire immune system response in real time.
So it's a crucial piece of the puzzle, but it's not the whole picture of what goes on inside a person.
Correct. In a real infection, you have blood flow, circulating immune cells coming and going, complex signaling across the whole body, things this model doesn't fully incorporate. Also, the study mainly looked at the early stages of infection, up to about 6 days. Tuberculous meningitis in people develops over a longer timescale.
So it gives us a really valuable look at that initial breach, how the foothold is established, but maybe not the full chronic progression of the disease within the brain.
Exactly. But understanding that initial step, that entry mechanism, is so critical. As Proust pointed out, knowing how they get in is key for treatment ideas, maybe even leveraging that leakiness. And Wilkinson's points about the bystander effect, helping bacteria get in and grow, and the sheer difficulty of treating brain co-infections are really highlighted by these cellular findings.
Okay, so wrapping up our deep dive today, the really big takeaway seems to be that Mycobacterium tuberculosis doesn't necessarily need a Trojan horse. It looks like it can directly attack and damage the cells of the blood-brain barrier, making it leaky. And then adding HIV to the mix seems to give the bacteria a serious advantage, helping them invade those barrier cells more easily, grow faster inside them, and ultimately cross into the brain more readily, possibly even through these subtle bystander effects.
It's a vital piece of knowledge for understanding how tuberculous meningitis develops, especially for people living with HIV. It shifts the focus towards these direct bacterial actions on the barrier cells themselves.
It really makes you think about the constant battle between pathogens and our defenses. The brain's protection is so sophisticated, but figuring out exactly how invaders like TB find these chinks in the armor and how other infections like HIV can subtly help them, that's the real challenge if we want to protect this absolutely critical organ.
It does make you wonder, doesn't it? What other diseases might be using similar, perhaps unexpected strategies to get past the body's defenses?
Thanks for tuning in to the Health Headliner podcast by Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.